In this video, we will discuss about mKRAS affect this tumor microenvironment through lipid metabolites. So, because of the mutations in the KRAS, it will results in the release of the lipid metabolites in the tumor microenvironment, and these lipid metabolites helps in the immunosuppression. At the same time, it will also helps in these pro-tumorigenic conditions. So here the effect of these lipid metabolites is shown on three different things. That is, first one is the CD8 plus T cells, and second one is the CD4 plus FOXP3 plus regulatory cells, and third one is this M2 macrophages. So we will start with the CD8 plus T cells. So this free fatty acid, when the CD8 plus T cells interact with this free fatty acid, because of the change in the release of the lipid metabolites from the mKRAS mutated cells, so this free fatty acid will cause upregulations of this immune checkpoint receptors, that is PD1 receptors on the CD8 plus T cells, that can interact with its counter PD1 ligand on the tumor cells to suppress the immune response. At the same time, it will also cause the downregulations of the lytic granules, that is granzyme, and also this IFN gamma, and overall it will result in this reduced cytotoxic functions, and since cytotoxic function goes down, it will relatively increase the pro-tumorigenic properties. The next thing is about this CD4 plus FOXP3 plus regulatory T cells. So here also, when they come and interact with this free fatty acid, then there is a change in the pathway occurs in a way that this lactate will be released, or this free fatty acid will result in this lactate release pathway, and this change in the pathway will also has a role towards this high T regulatory cells. These regulatory cells' main functions is to suppress the immune response, and since the immune response is suppressed, it will proliferate. It will help in this pro-tumorigenic environment. And this free fatty acid, then now this along with this free fatty acid, the KRAS will also release the cytokines, that is CCL2, and both of these free fatty acid and this CCL2 cytokines helps in this M2 macrophage polarizations. So what does free fatty acid does is it will reduce the M1 markers. At the same time, it will upregulate the M2 markers, such as this STAT6, and these things usually happen. These things usually happen through this PPRA by gamma to STAT6 upregulations, and so this is and these M2 macrophages that we have known, and we have seen this in multiple papers, where the M2 macrophages has this pro-tumorigenic properties, that is, it will prevent the tumor cells from the immune attack. At the same time, it will also helps in this tumor proliferations. So how this CCL2 works is that this KRAS mutations will help in this PPRA delta ligands to PPRA delta activations. It interacts with this PPRA delta PPAR delta ligand to cause this PPAR activations, and this PPAR delta activations will cause the release of these cytokines, that is CCL12, and this CCL12 then interact with this CCR2 axis to helps in this macrophage polarizations towards this M2 type. And in terms of the CD8 plus T cells, not only this lytic granules, that is granzyme released, but the cytokines that helps in this tumor killing or immune response, they also get reduced once the PD1 interacts with this ligand on the tumor cells. And for this for CD4 plus FOXP3 T regulatory cells, then it will has the role towards these lactic formations. So instead of this following the glycolysis pathway, it will go through our lactate formations, and that will help these T regulatory cells to suppress or inhibit the other activated T cells. So this is how this mKRAS mutations affects three different major ways through the release of the lipid metabolites. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.